Hi everyone, welcome back. So we're already at week 5, pretty much halfway through the term. So this week we're going to get into the beginnings of metabolism. So chapter 6 will go through a little bit of physics and talk about energy transformations and so forth. And then we're going to use that information to be able to see how chemical reactions take place within our body. And we'll also take a look at enzymes and how they speed up those reactions. We will also get into the beginning of chapter 7, which is cellular respiration. So that's this very important reaction that takes place in our cells to basically convert our food energy into a form of energy that our cells can use, ATP. And we'll spend all next week talking about that as well. Okay, so let's get into chapter 6. So with regards to chapter 6, we're going to first talk about what metabolism is all about. We'll get into some of the definitions of things like what free energy and endergonic versus exergonic and all that fun stuff. We'll take a closer look at cellular energy, ATP, and the role enzymes have in chemical reactions and how our body regulates the function of those enzymes. So let's start out by getting us familiar with this term metabolism. So metabolism is basically just the sum of all the chemical reactions that are occurring inside your body. So you might think of a child as having a very fast metabolism, which makes sense because they're having all sorts of chemical reactions occurring inside their bodies in order to grow, right? And so their metabolism will be a lot higher. And, typically they need a lot more food for the amount of body weight they have. And this versus somebody who's old, their metabolism is going to slow down as you age because you're not growing anymore. So you're not going to have those as many chemical reactions. It's more for maintaining your body than for growth. So when we're talking about chemical reactions that are occurring, a lot of times those reactions, we actually have multiple reactions that take place until we end up with a final product. Kind of like when we talked about cell signaling last week with that transduction pathway where you have one reaction causes another, that triggers another. Same thing with these metabolic pathways. So we start with some starting molecule and it might undergo a reaction to create a different type of molecule that then goes under another reaction that creates another molecule, and then finally a reaction takes place until we end up with that final product. So that's called a metabolic pathway, where we have multiple reactions taking place in order for us to get the final product. And what's interesting about this is that each one of those reactions is catalyzed by its very own enzyme. So enzymes are very specific in the form of what reactions will take place in their presence. Okay, so more terminology. We're going to get into the terms catabolism versus anabolism. So in a catabolic pathway, we're actually going to be breaking down complex molecules, and in doing so, that's going to release energy. So catabolic is releasing of energy, breaking down molecules into simpler compounds. Anabolic would be the opposite. So it's going to require energy in order to build complex molecules from simpler ones. So I kind of like to think of it as, catabolic. Think of, a kid who is running loose on a sandbox and just destroys everybody's nice sandcastles that they built. So that would be an example of a catabolic pathway, because the little kid is just destroying everything. It's releasing his energy and it's breaking down the poor sandcastles into their original sand, whatever it was before the particles. So that's just going to be releasing energy. It's taking things and breaking them down. Anabolic, think of the opposite. Think of the kids that were in the sandbox carefully building their sandcastle. So that takes time and energy in order to build that. So they're building the sandcastle from the smaller components, the sand grains. So that would be an example of an anabolic pathway. Okay. Okay, and then getting into the physics, energy. So energy is the capacity to perform work. So we typically just take energy and put it into one of two forms. It's either as potential energy or as kinetic energy. So potential energy is stored energy. So it's energy that has the potential to be used. Whereas kinetic energy is the energy of motion. So it's actually doing work. So you can think of, for example, an apple. An apple would be an example of potential energy because it's got all of this stored energy inside, all the fructose and monosaccharides and so forth, all that potential energy that could power your cells. So it's stored, all the stored energy that we just need to digest and break down, and then our cells can use it. Kinetic energy would be actual using that energy. So it would be that apple has been transformed into nutrients that our body absorbs, and then we use those nutrients in order to gain that cellular energy to actually be able to walk and move and so forth. So kinetic energy is the energy of movement. It's the energy of heat. It's the energy of light. It's anything that moves, light energy, heat energy, and so forth. So that's kinetic energy. Here's another way to look at it. So imagine, you are about to dive off a diving board into this lake on a nice hot summer day. So this diving board, okay, and this person waiting to go has a lot of potential energy inside of him. So he has all this stored energy, and then when he finally dives off into the water, that potential energy is going to be converted to chemical energy, or the energy of movement. Okay, so he's diving in. That energy that was stored inside of his muscles are now being used in order to dive into the lake. And then once he has dove into the lake, and it's just treading, just basically sitting there, then he has less potential energy because he used that energy to get into the water. Okay, and then climbing out, that's going to require some energy. So kinetic energy is going to be used to go back up and try it again. Okay, so let's get into the laws of thermodynamics. So a law in physics is basically like a scientific theory. It's something that has a huge amount of evidence to support it. And so it's basically as close to proven as we can get, but we don't use that word proven in science, right? So the first law of thermodynamics is basically, you can call it basically it's the law of conservation. So this basically states that the amount of energy in the universe is constant. So that means that energy cannot be created nor destroyed. It can only be changed from one form to another. It can be transferred from one form to another. So for example, you can see here this grizzly bear, he's snacking on, looks like a salmon. And so that salmon has all sorts of chemical energy that is being brought into his body, and then that energy is going to be converted into a usable form for him to do whatever bears do. But he's not creating energy. He's using the energy that was stored in the fish, in the proteins and the lipids and the carbs of the fish. So that nutrient energy is a form of energy, chemical energy, and he's going to convert it into cellular energy. So the energy is not increasing or decreasing. It's not being created nor destroyed. It's just changing from one form, chemical energy, to another form, cellular energy. Okay. Same with our power plants. Our power plants, they don't make money, or they don't make energy. They just convert it from an unusable form to a usable form. So think of coal-fired power plants. So the energy is stored in the coal, and then when the coal is burned, it can transfer that energy to turbines that can then convert that into electrical energy that we can be using. Okay, so energy cannot be created nor destroyed. It can only be transformed from one form to another. The second law of thermodynamics states that the conversion of energy from one form to another is not going to be 100% efficient. So that means there's always going to be an incomplete conversion. So instead of 100 units of chemical energy being converted into 100 units of cellular energy, that would be 100% efficient. But due to the second law of thermodynamics, each transformation that occurs is going to be accompanied by entropy or disorder. So that means that it's going to be an incomplete transfer. The rest of that energy is going to be lost, as usually it's a form of heat energy, which you can't use. So for example, the chemical energy that's stored inside this salmon, that all those calories that the bear is taking in, all that unit of energy that it's taking in, it's not going to be 100% used for movement. Okay, some of it, when it's being converted, it just gets lost as heat and it gets off through that. Okay, and there's byproducts that are resulting. Okay. So another way to think of it is like your car. When you put gasoline in your car, that's an energy source, right? And then your car burns that fuel in order to make your car move. But there's a byproduct there. It's not a 100% conversion from gasoline into the movement of your car. It's going to be releasing heat. And that's why, after you've driven a while, the hood of your car feels warm. So you're losing that heat that is an incomplete conversion. So let's start out, let's watch a video that kind of goes through the concepts of energy.
Energy is the capacity to cause change. Kinetic energy is the energy associated with motion. Energy that is not kinetic is called potential energy, and is energy that matter possesses because of its location or structure. Energy is neither created nor destroyed, but converted from one form to another. This property is called the conservation of energy.
The bow and arrow start out with low potential energy. As the archer's arm pulls back the string, kinetic energy from the arm's motion is converted to potential energy in the tense bowstring and arrow. When the bowstring is released, the stored potential energy is converted into the kinetic energy of the moving arrow. When the arrow hits the target, its motion ceases. If energy is neither created nor destroyed, where did the energy of the flying arrow go? Let's rewind and look at kinetic energy in more detail. When energy is converted from potential energy to kinetic energy, some of the energy can be used to do work, but some energy ends up as heat, a type of kinetic energy. Heat is the random motion of atoms and molecules. As the arrow flies through the air, heat is generated by friction between the arrow and air molecules. When the arrow strikes the target, all the energy becomes heat energy. The heat energy is rapidly transferred into the air and spreads out. Heat energy is a very disordered kind of energy. It has the highest amount of entropy, or disorder, of any kind of energy. Where do our muscles get energy to perform work, such as pulling back a bowstring? Our bodies use the chemical energy from food to perform work. Chemical energy is a form of potential energy. When your body breaks down food molecules, the stored potential energy from food can be converted to kinetic energy. The stored chemical energy in food is released in your muscle cells during the process of cellular respiration. Using oxygen, cellular respiration converts chemical energy from food to another form of chemical energy called ATP. Water, H2O, and carbon dioxide, CO2, are byproducts of cellular respiration. The potential energy of ATP can be converted to kinetic energy. Some of the kinetic energy is used to do useful work, such as pulling back a bowstring, but the heat energy that is generated cannot be used to do work.
Okay, so that video, gave us a good example of both, the difference between potential energy and kinetic energy, as well as, really defined the first law of thermodynamics and the second law of thermodynamics. Okay. So let's go ahead and continue looking at this idea of energy, specifically what is known as free energy. So free energy is energy that can do work. And we generally describe it as basically when the temperature and the pressure is uniform within a living cell. So you'll see this symbol throughout, the chapter readings if you haven't had a chance to look at it. So this symbol, delta G, just basically stands for that delta signal, stands for change. So in this case, it's the free energy. So that's taking the final state of the energy minus the initial state. So the change in that is going to be the amount of free energy. So that's basically during some sort of chemical reaction, it's the difference between that, the product energy state minus the initial state of that energy. Now, if we end up with a negative number, then it's considered a spontaneous process. Because if you think about it, do I have an example? Maybe I can write it here. Okay. So if we have, let's say, we are converting, I don't know, some sort of reactant A and B, and then we're going to make it into AB. Okay. So let's say that the final state, AB, is going to be, I don't know, I'm just going to throw a random number. Let's say it's a magical number of 10 units of energy, okay? And the initial state, let's say that it was basically 12 units of energy, okay? Again, this is just very simplistic. So if I take that final state of 10 amount of energy and I subtract minus 12, I'm going to end up with a negative number. So that means that the initial state has more energy than the final state. So no energy needed to be added to this reaction to cause it to occur. So that would mean it's spontaneous. If we had a positive free energy, then it does require energy in that state. So for example, if my final state was, I don't know, 15, so this would be the final state. I'm just going to write it in parentheses, 15. But my initial state was, say, 12, okay? I still need energy to make sure that the reaction takes place because there's not enough to make 15. So that would be an example of a reaction that requires energy input. Okay, so spontaneous process, look for the negative free energy, so no energy input, and then other processes require energy input. Okay. So let's bring it down to some examples. So here we have this person is about to jump off of a platform, okay? So in this case, there's a lot of stored energy inside, okay? So this amount of stored energy is potential energy. And then we're going to add gravity, okay? So gravity is the pull of the earth. So that's even more energy that's added to the initial state. So in this case, there's going to be a lot more energy available at the beginning. because there's higher amount of free energy at the beginning than at the end. And since there's less energy at the end, that means that there's going to be less free energy, so there's going to be no energy input into the system. So it's easy. Basically, when you jump off a platform, that's easy, right? Because it doesn't require that much energy. Another example would be diffusion. Remember how concentration gradients, so simple diffusion, you have molecules have a tendency to go from higher concentration to lower concentration. So this doesn't require any energy input, right? We're not talking about active transport, we're talking about passive transport. And so the energy capacity in this state is higher than this state. So it's going to, require no energy input for this reaction or this movement to take place. Okay. And then here we have an example of a chemical reaction where this complex molecule is starting out at a higher state of energy than the products, and so it's not going to require any energy for it to break apart. Okay, so the free energy is going to be higher in this state compared to this state, so we're going to end up with a negative free energy. Okay, so if we go back and talk about the chemical reactions, we have basically, we have two types of chemical reactions in relation to the amount of free energy. So the first type is called exergonic. So exergonic means that there is a negative free energy. So there's so much energy in the reactants that energy will actually get released during the reaction. Okay, so we start out with high amount of free energy, and then we end up with low amount of energy, right? So that energy, the free energy, is going to be a negative number, okay, because it's getting released. So this is an example of a spontaneous reaction. So think of burning wood, for example. So once the wood is ignited with that heat source, it's basically that heat source, the carbon inside that wood is has a lot of energy. And so it's going to basically have higher amount of energy than the ash that's the end product after you burn the wood. So that energy gets released, and that's energy gets released as heat and light, right? Cellular metabolism is another example of an exergonic reaction. So we'll talk more about that when we get into cellular respiration. So we're going to have glucose molecules that are very energy rich, and they're going to get broken down and release energy in the form of ATP that we can be used. So exergonic, think releasing energy. Okay, so free energy will be negative. The opposite, endergonic, we need energy. Okay, so we need that energy input. So endergonic, think energy input is required. And that's because there's not enough energy in the reactant to produce the product. So we require an extra bit of energy to drive that reaction. An example of that would be photosynthesis. So in photosynthesis, the plants are taking in carbon dioxide and water, and they're making glucose from it, right? But they need light energy from the sun to drive that reaction. So that would be an example of an endergonic reaction because it requires that light energy to convert that carbon dioxide and water into the glucose molecules. So endergonic energy is required, therefore it's not spontaneous. Okay. Here's another example. If we have an isolated hydroelectric system. So here we have more energy behind this turbine than down here, right? So there's a lot of stored energy, and then that's being converted into the movement of water. So as that reaction's taking place where the water is moving from this high point to low point, okay, with the help of gravity, eventually gravity is going to run out, right? Because eventually the water is going to be at the same level. So in this case, the free energy is equal. We don't have any extra energy that's being released, and so it's stabilized. If we create an open hydroelectric system, we can keep the energy, the amount of free energy at a negative number if it's a multi-step process. So you can see how if we keep adding water to the system, okay, that's going to keep the turbines flowing, releasing energy. And then if it's set so that we can keep using that extra energy from gravity, that will keep the free energy at a lower level. So what does this have to do with biology, these turbines? Imagine instead of a turbine, we have an enzyme. Okay, so these are different enzymes catalyzing the reactions. So we're going from one reactant to form a product that will become a reactant to form another product, and so forth. So this could be an example of a metabolic pathway where we have an enzyme is going to keep the free energy at a negative number. Okay. So here's a question for you. Cellular respiration is an exergonic process. So remember that energy must be conserved. What becomes of the energy extracted from food during cellular respiration? So what is the goal of cellular respiration? What do we need? Okay, so we're converting what form of energy to what? So we're taking in our food particles, and that's a chemical form of energy, right? And we're making cellular energy or ATP from it. Okay, so that's the goal, is making ATP. So that falls under that first law of conservation or thermodynamics. So we're converting one form of energy to another, right? We're taking chemical energy from the food, and we're creating ATP from it, which is a form of potential energy that our cells can use. But during that process, it's not based on that second law of thermodynamics, it's not going to be a 100% conversion. So in addition to producing ATP, we have heat that is lost. Okay, so that energy that is extracted from the food becomes ATP, and it produces a little bit of heat as well. So this brings us to the mighty ATP molecule. So we're going to take a look at what ATP is and why it's so important for our bodies and our cells.
ATP. We love it. You'll find ATP in so much of our science art. Whether it's a GIF of a mitochondrion or cell transport, or a comic about cellular respiration or fermentation, you'll see ATP mentioned. So why the big deal? Why is it all over the place? Many times students will get it in their mind that it is an energy currency of some kind. When I first started studying biology, I noticed that in textbooks, it's often represented like this starburst thing or thunderbolt. And I guess in my mind I imagined it was like some big blast of energy to help the cell do things. And by do things, I mean that we need ATP to do many cellular processes. Examples include active transport, such as when a cell is trying to move something against its concentration gradient, or its role in muscle contraction with the actin and myosin cross bridge. We need another video for that. ATP is critical for many types of cell signaling. You need your cells to be able to communicate. Those are all just some examples. But what is ATP? How do we get it? And how does it work? Those are the basics of what we're going to focus on in this short video. So what is ATP? If you remember the four major biomolecules, ATP would fit in with nucleic acids. Yes, like DNA and RNA. ATP is a nucleotide derivative, so it has those three parts you'd see in DNA or RNA nucleotides: phosphate, sugar, base, but it actually has three phosphates. ATP is short for its full name, adenosine triphosphate. This fancy name is helpful as it tells you that it contains the nitrogenous base known as adenine and three phosphates, hence the tri in adenosine triphosphate. Its sugar is ribose. How do you get ATP? All cells need ATP, and so they need processes that can be used to generate it. But the process can differ. It might involve oxygen, such as aerobic cellular respiration, It might not involve oxygen, such as anaerobic respiration or fermentation. During cellular respiration, plants break down the glucose they made from photosynthesis to make ATP. During cellular respiration, animals break down the glucose they consumed to make ATP. And it's not just plants and animals. Bacteria, fungi, protists, and archaea, they all need to make ATP. We have a video on cellular respiration and another one on fermentation that can be helpful to understand the process. But one thing that we do want to mention about making ATP is it's important to understand it is part of a cycle. With the ATP cycle, you have ATP, which can be hydrolyzed, releasing energy and losing one of its phosphates in this process. A process like cellular respiration can provide the energy needed to add a phosphate to ADP in order to regenerate ATP again, which is important as ATP can be used quickly. This brings us to how ATP is able to work. So how does ATP work? It's not just about ATP being hydrolyzed and releasing energy, it's more than that. Okay, honestly, it's more than our short video can go into, which is why we provide some further reading links, but let's look at some basics. So when ATP is hydrolyzed, meaning here it involves the addition of water, it's not really that the bond between the second and third phosphate itself is a super strong bond. It's actually more that the bond between the second and third phosphate contributes to this ATP being unstable. These phosphates with their negative charges don't like being arranged like this. The change from ATP, losing its third phosphate, to become the more stable ADP is an exergonic reaction and releases free energy. A popular example for understanding ATP is to use the spring illustration, like a wire spring. Consider how you might compress the spring. ATP would be modeled by that compressed spring, and then you would let it go until it just goes into this relaxed state, which would be represented by ADP. When ATP is hydrolyzed, if the energy was just released, it will likely not be useful for a cell if it's not actually coupled to something that needs it. Thankfully, the energy release can be coupled to endergonic processes that the cell needs to do. This can occur when the phosphate from the ATP is transferred to a molecule that is going to be acted upon. For example, this cell transport protein here is supposed to move some kind of molecule against its concentration gradient. Recall that if it was passive transport, these molecules would be moving from high to low concentration. But in active transport, thanks to ATP, this protein can move them against the gradient. When the phosphate is transferred to this protein, we say the protein has been phosphorylated. Sounds powerful. We can say in our example that this protein is more reactive and less stable in this form, this phosphorylated intermediate state. When it reverts into its original, more stable shape, it can assist in moving them the other direction. So, from marveling at the beating of a single cilia hair, or chromosomes being separated in cell division, or binding the correct amino acid on the tRNA, I could go on. We hope that little ATP symbol will mean something every time you see it. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Okay, so that was a good introduction into this, magical molecule known as ATP. So we're going to be using this molecule ATP, which again stands for adenosine triphosphate, to help explain how cells do their work. So there's three main kinds of work that a cell does. It could be chemical work, it can be transport work, or it could be mechanical work. And ATP is the energy source for each one of these types of reactions. So let's take a closer look. Here we have ATP, that magical molecule that the Amoeba Sisters talked about. And basically in this process, remember, through that process of ATP, it's going to undergo a reaction where the phosphate is going to be broken off of the ATP molecule to form ADP. And that burst of energy is going to allow that reaction to take place and the products to be formed. So here's that example in chemical work. So you can see how that phosphate group is going to be basically used to allow this reaction to take place, where that the two reactants form the single product. In transport work, we had that as an example in the Amoeba Sisters video. So as ATP undergoes the reaction, the phosphate group can then trigger the protein, transport protein, to change its shape to allow something to happen. Okay, and then we end up with this ADP as a byproduct or as the end result. Mechanical work, here's an example of a motor protein with some actin, and that's basically, you can see how the reaction, the ATP molecule, as it releases that phosphorus, it's going to cause a change. That phosphate group is going to add to the motor protein to cause a change, and that's going to cause movement. Okay. So we can see how ATP is going to be used in order for all of these different reactions to occur inside of our body. But what is interesting about it is that ATP is going to be formed into ADP plus a phosphate group, and then can be combined to form ATP again, and then that can be used to form ADP and phosphate releasing energy, and then that can be combined to form ATP again. So that's called a energy coupling. So the use of an exergonic process to drive an endergonic process. So most energy coupling in cells is mediated by ATP because of that process. So let's take a closer look at ATP, the molecule. It is a nucleic acid in the sense that it contains the nitrogenous base, adenine, plus we have the sugar ribose, okay? And in addition to ribose and adenine, we have 3 phosphate groups. Okay, so you can see how the phosphate groups are arranged. They don't really like that arrangement too much, so that way it does make it easier for one of them to leave. So here we have, in this case, we have our adenosine triphosphate. It's got 3 phosphate groups, and then basically this is going to undergo a reaction. In this case, a hydrolysis reaction, so we're adding water to break the bond. And since we're adding water to break the bond, is this an exergonic or endergonic reaction? So remember, exergonic, we're releasing energy. So there you see the energy is being released. Okay, so this would be an example of a spontaneous reaction. Okay. Now, we can use this in all of our different reactions that occur inside of our body. So here, let's look at glutamic acid conversion to glutamine. So we have glutamic acid plus ammonia. Now, the amount of free energy is going to be a positive number. So it requires energy for these 2 molecules to react to form glutamine, okay, because we have a positive amount of free energy. So this is not going to be a spontaneous reaction. But if we couple this reaction with ATP's reaction, we can use that free energy that's generated, or that negative amount of free energy, that energy that's lost through that spontaneous reaction, and we can use that to power this reaction. So you can see how the 2 reactions are coupled. So here we have glutamic acid plus ATP. So ATP is going to undergo its reaction to form ADP plus that phosphate group. That phosphate group will attach to the glutamic acid to form to be phosphorylated. And then now it has enough of that, a higher amount of energy in its initial state, a lot more energy than in the final state. So that's going to allow for this reaction to take place now. So we're getting the energy that was released from the ATP, and that's going to be used to drive this reaction. Okay, so that's where the coupling takes place. So these two molecules together, okay, it's going to require 3.4 kilocalories per mole in order to form this product, okay? So there's basically, that's, you know, we don't have enough energy to perform this spontaneously. But the energy that is released from ATP, look at all that free energy. So we can use that, so we have an a net free energy as a negative number. So the addition of that ATP into this reaction will help create an exergonic reaction from it. Here's an example with ATP being added to that transport work. So you can see here's our transport protein. And then through that process, ATP is basically broken down, releases that phosphate group, which will attach to the protein. And that's going to cause that reaction, that change of state. And then we already talked about how the mechanical work one, where we have the motor protein. That motor protein is going to use the phosphate from the ATP being hydrolyzed, and that's going to cause or provide enough free energy for it to react or move in that case. So we coupled both of those chemical reactions. So exergonic is going to be coupled with endergonic. So remember, exergonic energy gets released. Endergonic energy is inputted into the system. So here we can have energy from catabolism. So remember, catabolism is breaking down you know, a big molecule into smaller forms, and that's going to release energy in the process. And so that release of energy from that catabolic reaction or exergonic reaction can help ADP and that phosphate group reform into ATP. So there's enough energy to bring these together, okay? And then when ATP is going to undergo hydrolysis, where it's going to break apart, so that's an anabolic reaction, right? And that will be an example of a reaction that is going to release the energy. And so that released energy can then be used in some sort of energonic reaction of some sort of cellular work that's taking place to allow that reaction to take place. Okay, so you can see how the exergonic reaction is coupled with the endergonic reaction with the help of the ATP molecule. So explain how ATP transfers energy from exergonic to endergonic processes in a cell. Okay, so remember those exergonic reactions where we have, I don't know, product AB is converted to A plus B, that's going to release energy, and that energy source can then convert ADP plus phosphate into ATP. And then from there, that ATP can then be hydrolyzed back down to ADP and phosphate, releasing energy that will provide enough free energy for an endergonic process, or a negative state of energy, I should say. Okay. All right, so we talked about ATP as, you know, can be coupled in reactions to allow them to take place. And that's very important for our metabolic activity. But we also need to speed up these activities. And so this is where enzymes come in. So enzymes, they catalyze reactions or speed them up. So they increase the speed of a reaction to take place. And what's interesting about enzymes is that they are never used up. So you can use an enzyme over and over again. It just gets recycled over and over again. And what it's going to do is, you see in this reaction, take a look at this reaction here. So we have a reactant and we have a product. Notice the amount of energy, the amount of free energy. So in this case, the reactant has it has more energy than the product. So this is going to, undergo a reaction. But there's this activation energy barrier that's in place. And so you would normally think, hey, this reaction should just take place because there's a lot of energy on this side of the equation than that side. But there's this barrier that's there that we have to get over first. So we do require a little bit of energy to cause this reaction to take place. So one thing that can be added is an enzyme. So the enzyme will actually lower the amount of activation energy that's required for this process to occur, this reaction. Okay, so let's watch a video to kind of explain it.
If exergonic reactions occur spontaneously, what keeps molecules from breaking apart and cell chemistry from racing out of control? For any reaction to occur, even a downhill reaction, some energy must be added to get the reaction going. This energy is needed to break bonds in the reactant molecules. The energy needed to start a chemical reaction is called the energy of activation, E sub A. This required energy input represents a barrier that prevents even energy-releasing exergonic reactions from occurring without some added energy. How does a living cell overcome the energy barrier so that its metabolic reactions can occur quickly and precisely? A special kind of protein called an enzyme is the answer. An enzyme serves as a biological catalyst, increasing the rate of a reaction without being changed into a different molecule. An enzyme does not add energy to a reaction. Instead, it speeds up a reaction by lowering the energy barrier. An enzyme is very selective. Its three-dimensional shape allows it to act only on specific molecules referred to as the enzyme substrates. As the substrates bind to the enzyme's active site, they are held in a position that facilitates the reaction. This takes less activation energy than the unaided reaction. Products form and are released. The enzyme emerges unchanged from the reaction.
Okay, so that was a good introduction. So, we always use like dehydration, synthesis reactions, and hydrolysis reactions, where basically we have some sort of, polysaccharide, and it undergoes a hydrolysis reaction to form the monosaccharides, for example. And this should be an exergonic reaction, right? Because it should be spontaneous. We have this large molecule that has a lot of energy inside, and then it's being broken down into smaller components with less energy. And so there would be a negative free energy in that equation. But we still need to start the reaction, okay? So the carbohydrate is just going to sit there. That polysaccharide is going to sit there, but we need to be able to break the bonds initially in order for it to undergo that reaction. And so that's where that energy of activation or activation energy is required. So that's that little bit of energy that's required to start the reaction. And so the enzymes are there to lower that amount of energy.
Yep.
So here you can see that if we don't have the enzyme, let's say we have, some sort of glucagon, a polysaccharide over here, and we need to make some glucose from it. We need to break it down into the monosaccharides. So in order for that glucagon to be broken, the bonds to be broken and released, we need a little bit of energy. Okay, so that's that energy of activation or activation energy that's required. If we add an enzyme, that activation energy is lowered. So that's going to speed up the reaction because you don't need as much energy to initiate the reaction. Okay. So let's watch an Amoeba Sisters video to kind of go over what enzymes are.
Is it odd to have a favorite protein? Well, I don't think so. Probably because my favorite protein happens to remind me of one of my favorite childhood games, Pac-Man. If you haven't played Pac-Man before, then chances are we're much, much older than you. But now you can play it on Google. Just Google Pac-Man. It's a Google Doodle. Anyway, I digress. But see, in Pac-Man, you have this little character. It goes around, finds these pebbles, and the pebbles fit right into it. Well, a lot of illustrations that you will find of enzymes, they happen to look, at least to us, a lot like Pac-Man. I remember P for Pac-Man and P for protein. Most enzymes are proteins. Now, in the game, we mentioned there are these little pebbles that Pac-Man goes after. Well, enzymes have a specifically shaped area called an active site where items can bind. The items are called substrates. It's very specific binding because the active site is specifically shaped for the substrate that binds there. Very specific. So what happens when a substrate binds an enzyme? Well, usually the substrate is held there with some weak bonds because it's not going to stay there forever. And something called induced fit will happen, which means the active site can change its shape even more to bind that substrate, like an enzyme substrate hug. The enzyme can either build up or break down the substrates that specifically bind to it, and we call the resulting item the product. An enzyme has the ability to really speed up reactions. Reactions that technically could happen on their own, but with the help of enzymes, they can be sped up to make processes effective for life. Let me give you a great real-life example. The enzyme lactase. Another really cool thing about enzymes is that they often end in ASE, like lactase. Many sugars, on the other hand, end in OSE, and lactose is an example of a sugar. Lactose is a disaccharide, meaning it contains two sugar molecules bound together. We don't actually digest it so well in that form. It's big. The enzyme lactase has the ability to break lactose down into smaller parts that our body can digest. And this is a lot better option than waiting for a chemical reaction with lactose to happen spontaneously. Now, with the lactase enzyme, lactose can be broken down quickly and digested. Now, there are some people that do not produce enough lactase. They can be what we call lactose intolerant, which means that if they consume foods that have lactose, such as milk, it can make them sick. They can't break the lactose down efficiently without lactase. In that example, one thing to point out, lactase, the enzyme, can break down a lot of lactose, the substrate. The lactase doesn't get used up in the reaction. It's still there. We call enzymes a catalyst because they can be used over and over and over and over in the reaction. By the way, your digestive system uses all kinds of enzymes. You have lipase, and that breaks down lipids, which are fats. You have amylase, which breaks down starch. You have protease, which breaks down proteins. So as you can see, the digestive system is very involved with enzymes. Another thing to point out is that enzymes don't always work alone. Sometimes they get some help. Some often underappreciated but essential little helpers are called cofactors and coenzymes. They may bind to the substrate or to the active site. They help the enzyme do its job of building up or breaking down substrates into products. Now, you didn't forget our Pac-Man analogy yet, right? In the game Pac-Man, there are these ghosts. And when they touch the Pac-Man, it makes this sound. It's like... And the Pac-Man shape gets all distorted in the process. So what does this have to do with enzymes? No, there aren't ghosts around. But enzymes do have certain ideal conditions that they like. For example, an enzyme that is in your stomach would have an ideal pH that is very acidic because the environment in your stomach is very acidic. Different enzymes have different ideal pH and temperature ranges. If an environment changes out of an enzyme's ideal pH or temperature range, then something that reminds me a lot of that horrible sound I tried to make can happen. The enzyme becomes denatured. That means its shape becomes distorted. It can no longer bind to its substrate. It can no longer work correctly. It is finished. Well, that's a dramatic end to enzymes. Keep in mind that if you have an interest in this topic, many medical researchers have a large focus on enzymes. Enzymes regulate a lot of the body processes, and many diseases can involve specific enzyme production, or the lack of it. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
All right, So that was a good introduction into basically the structure of an enzyme and how it's used to catalyze reactions and some of the potential issues with enzymes, okay, as far as becoming denatured. So let's take a closer look at an enzyme, and then we'll call it a day and complete the rest of the lecture in the next video. So in enzyme structure, we have to take a very close look at the enzyme. Now, enzymes, as the video talked about, are mostly proteins, right? And if you recall, wow, that was quite a while ago when we looked at the macromolecules and the primary structure versus secondary structure versus tertiary structure of a protein. And remember how the primary structure is going to determine the secondary structure, and that's where those H bonds come into play, and then you got the tertiary structure where you got like the R groups in. So that's going to allow this protein to have a certain shape, and that unique shape has an area that has a specific spot where some sort of substrate can bind to it. So that spot, based on that three-dimensional shape of the protein, is called an active site. Okay, so that's that special little spot that allows the protein to carry it, the substrate. And it's very specific, okay? So it's specific to only this type of substrate generally. And so only that particular substrate has that ability to bind to it. And then once it binds to it, we saw how in the video it kind of creates a little hug. So it kind of the protein even forms more around it. So that's that idea of induced fit. And so this forms something called an enzyme substrate complex. So we have the enzyme here and we have the substrate attached to its active site. Okay. So let's see how this works. So here we have a couple of different substrates, and those substrates, they fit perfectly in the active site of this particular enzyme, the purple thing. And so these substrates are going to have a weak bond with the protein, and that's going to cause them to be able to react together in this enzyme substrate. So this is now lowering the activation energy of the substrates in order for the reaction to occur. So here they're converted to some sort of product. And now that the reaction has taken place, then those weak bonds that held the substrates, now products, to the active sites are broken, and we end up with our products. Okay, so we've seen a reaction take place. The substrates are now products, they've changed. And we had that enzyme involved in catalyzing that reaction by lowering the energy, activation energy. But what happens to the enzyme? Well, it gets recycled. Now there's nobody occupying the active site, so it can get ready for the next set of substrates to bind to it. Okay, so it can be used over and over again. And then the video talked about how since that protein, that enzyme, is such a specific shape, that any changes in that shape can cause the enzyme to no longer be able to catalyze a reaction because it changes that active site. So if the active site is different, then the substrates can no longer bind to it. So that's that term denature, where the enzyme has changed its shape and thus it doesn't work anymore. So we can think of that as from the protein structure level. So the primary structure, the secondary structure where those H bonds are involved, we have these bonds that are occurring in that secondary structure, but if there's some sort of change in the environment, that can cause some of these weak bonds, those H bonds, they could break, and then now you've had a change in the protein structure, so it's denatured. So what are some things that can cause an enzyme to become denatured? Temperature is certainly one of them. The majority of our enzymes in our body, they optimally work around 37 degrees Celsius, which happens to be our body temperature. Hey, I wonder why. And then we can think of what happens when we get really, really cold and we suffer from hypothermia. Hypothermia is when our temperature goes down. And since the temperature has gone down, it's going to, slow down the molecules, so the reactions are going to be slowed just naturally from that. If we go the opposite direction, if we increase the temperature of our body, it's called hyperthermia, we can end up having our temperature go beyond what is optimal for the enzymes. And then that heat, that added heat, can change the structure and denature the proteins. So this can occur when we have something like heat exhaustion, which then could lead to something called heat stroke. So in heat stroke, that means that our body is no longer capable of keeping our temperature at a cooler rate. And so our body just suddenly just goes into shock, and it starts kind of shutting down its organs because it can no longer support, basically those enzymes are starting to denature because the temperature is just too high. So overheating can cause a lot of issues, especially it can lead to brain damage and even death because of the denaturing that can occur of the enzymes. Salt concentration can also change the structure of a protein. So high salinity environments can cause a change in basically how an enzyme functions. Another one would be changes in pH. So you think of pH in our body is usually around 7, a little bit higher, a little bit lower, depending on where we are. But one area of our body, our pH is around 2. It's very acidic, so inside of our stomach. So those gastric juices, they contain enzymes such as protease, peptide. I can't remember. Pepsinogen. I can't remember. But anyway, it's the one that breaks down proteins. I think it's protease. But anyway, it can survive in pH 2. That's its normal environment, that particular enzyme, because it's breaking down your proteins into amino acids. If it suddenly gets into your your small intestine, where it's a more alkaline environment, it's going to denature. Okay, so that commonly occurs. So pH is important, and then certain chemicals. So there are a lot of ways that we make some of our medicines have to do with denaturing enzymes in the pathogens that are infecting us. So, for example, antibiotics can inhibit certain enzymes that help build the cell walls of the bacteria that aid in protection. You can think about pesticides that we place on our plants. A lot of those, they block certain they cause a denaturing of the enzymes that make it difficult for the animal, that insect, from being able to transmit nerve impulses. So it ends up dying from that. So certain chemicals can denature enzymes as well. So here's just an example showing how we have 2 different enzymes. Pepsin, that's what it was. So that's a stomach enzyme. I knew it was something similar to that. It doesn't have the ACE at the end, sorry. But you can see its reaction to different temperatures. So in our typical enzymes, we'll get to the pepsin in a second. So a typical human enzyme is around 37 degrees Celsius. And it can, tolerate cooler temperatures, but it eventually will begin to denature if the temperature is too high. Other organisms like bacteria, they can survive warmer temperatures just fine. So like a thermophile is a type of bacteria that can live at extremely high temperatures. So think of, hot springs environment where you end up with basically just bacteria in there and archaeans, some prokaryotes. Not much else can survive there because it's too hot. And that is because their enzymes are actually working optimally at a very high temperature. Okay. So each, enzyme has its optimal temperature. If we deviate too much beyond it, it can cause it to denature. Same with the pH. So here we have a stomach enzyme versus an intestinal enzyme. So pepsin right around pH 2 is optimal. In the small intestine, we have trypsin that will help break down some of our nutrients, and that works at a pH of 8. So you can think, okay, well, trypsin's not going to survive in the stomach and pepsin's not going to survive. Well, it can't really survive. These are not cells, but it'll denature if it's placed outside of its optimal pH. Okay, so trypsin will not function in pH 2 like pepsin. Pepsin will denature if it's placed in the small intestine. All right, and then we saw from the video that there were those things called cofactors. So cofactors are non-protein enzyme helpers. Now, you might remember, you're supposed to have your multivitamin because it has the daily dose of your vitamins and your minerals. But how do you fit everything that your body requires into a small, tiny pill? Okay, those are actually coenzymes, okay, or cofactors, I should say, because they can get recycled just like the enzymes themselves. So we just need them in minute quantities. So if it's an inorganic metal, like a mineral, I think, zinc or something like that, that would be an example of a cofactor. That I forget what they called it, but so that's basically a mineral. So minerals are inorganic. The vitamins would be organic, so they would be considered a coenzyme. So these are examples of cofactors that will help other substrates bind to the active site. So most vitamins act as coenzymes and are the raw materials of which coenzymes are made. So we just need them in minute amounts. Okay, same with the minerals. All right, so explain how an enzyme speeds up a specific reaction then. So remember, in order for that reaction to take place, we have to lower that energy activation. And so the enzyme is going to allow for the substrate to bind to that active site, which will then force the substrates to be able to react. And so by lowering that activation energy. And so that's going to allow the reaction to take place, speeding it up. Okay, so it's lowering the activation energy needed. All right. Okay. So I'm going to go ahead and stop right here for this video. In the next video, we're going to finish up enzymes. We're going to talk about how enzymes can get regulated. And then we will, so we'll finish up chapter 6, and then we're going to just briefly start chapter 7. We're going to do 7.1, so just the beginning part of chapter 7. Okay, so I will see you in the next video.